Okay, good morning and welcome to the final lecture of Evolutionary Robotics. We are going to spend most of today talking about evolving bodies and brains for biological robots, otherwise known as the Xenobots. Before we do that, we will finish off lecture 23 and our discussion of differential robots. Before we do that, just a reminder and orientation for the final project. Super important. You get both of your products submitted to Brightspace by 11:59 p.m. This coming Monday night. There's a link there that explains exactly what you're submitting. You should see in Brightspace now there are two additional submission portals for you. You're dropping a link into both. One is a link to the PDF for your written report. The other is a link that points to a public 2.5-minute silent YouTube video. Yeah? Okay, that's Monday. Tuesday, as I've already mentioned, we'll all be here bright and early. I will, maybe today, maybe tomorrow, I'll draw up the list of presentation orders, and I will drop the link in here so you can see exactly when you're presenting. If you're presenting later in the morning on Tuesday, please be here along with everyone else at 7:30, and please stay till the end. I know everyone is very busy next week, but please, you're going to see some amazing projects. Stay for the duration. I will supply you with sufficient sugar and caffeine. Deal? Awesome. Okay. Any other questions about the final project? Last chance. Going once, going twice.
The video is recordings of the simulations evolving. Is that what it should
be? What videos do you want to see? Do we want to see inside that short video? We want to see whatever videos you want to show us, but the teaching assistant and myself are looking for those videos and whatever other visualizations you include to collectively answer two questions. Question number one, is your evolutionary algorithm doing anything? If you show us a video of a random robot and an evolved robot, all of us should be able to see within a few seconds that the evolved robot is doing a significantly better job at climbing the ladder, jumping the gap, carrying something with its partner. Whatever it is, it should be immediately obvious. And we want to see some quantitative evidence. We talked about this last time, fitness curves. Convince us beyond a shadow of a doubt that your evolutionary algorithm is doing useful work on your behalf. Question one. Question two, you're going to then show us two different variations of your final project, A and B, and you're going to show us videos and quantitative data in some form to answer the question, is A doing better than B? Is B doing better than A? Or is there clearly no difference between A and B? The fourth option, which is, I don't know whether A is better than B, B is better than A, or there's no difference, that's the one you want to avoid. The way to avoid it is collect enough data and present videos and quantitative data to convince us. Any other questions about the final project? No? Okay, all right, so back to differential robots for a moment. Again, sorry, just a little bit of reorientation here. We're looking at evolving bodies and brains, which is really what evolutionary robotics is all about. This is what makes it different from all other branches of robotics. We're interested in optimization methods that we can broaden such that they can automatically tune both the brain of the robots and their bodies as well, as happens in nature. We went all the way back to the mid-90s to see the first attempts at this using rigid robots. Then we looked at some recent work over the last few years on using similar techniques to evolve bodies and brains. In lecture 23, the thing that we're changing is we're removing the evolutionary algorithm and we're plugging in a different optimization method called gradient descent. It is more complicated. It's less intuitive than evolutionary algorithms, but its benefit is it removes the randomness that exists in evolutionary algorithms. In artificial evolution and biological evolution, it's a lot of trial and error, and unfortunately in nature and our robots, some of them suffer because of that. They're the wrong fit for whatever the solution is we're trying to solve. Gradient descent removes the guesswork. There is no more randomness. So how does this work? We were trying to develop an intuition for gradient descent applied to robots by applying gradient descent to the most, hopefully at this point, the most familiar robot in this course, Bratenberg vehicle. So just a reminder, there's a light source emanating from the front left of this robot at this moment in time. That light falls on both light sensors, but it falls on the left one more than it does on the right. We're going to introduce a variable x, which we're going to fiddle with in a moment, which is the horizontal position of the left sensor. Okay, so we've got x. There's lots of other variables, but we're going to just pick a few. We then have theta, and theta is a single number, a single floating point value, which is the weight of the synapse connecting this input neuron to this output neuron. This output neuron, whatever that output neuron's value is dictating omega, the angular velocity of the wheel. How fast should the wheel turn? So, as you'll remember with Bratenberg vehicles, at this moment there's more light falling on the left. If theta is if these two weights, this theta and this theta, are both equal, that means that this angular velocity is going to be greater than this angular velocity, and it would turn towards the left. The Bratenberg vehicle is so simple that we can dream up what good thetas should be. We're going to assume, and you've seen in this course many times now, that once the robot starts to get more complicated, we can't do this anymore. We need to go searching for good values of thetas, the brain of the robot. And in some cases, we also want to go looking for variables that describe the body of the robot. Okay, so we have x, theta, and omega. We want the robot to do this. We want it to turn to the left, so we know what we want. We're going to pick some random values for x and theta, as we usually do at the beginning of optimization. We don't know what the optimal values of these variables should be, so we're lazy. We just pick some values at random. We run the simulation forward. We're going to refer in lecture 23 to the forward pass. Forward meaning we're taking information from the physics engine, in this case light and gravity and everything else. We get sensory information, that's the sense part. We flow sensory information through the neural network controller, that's the think part, sense think. The values arriving at the output neurons dictate how the robot acts, that's act, sense, think, act, that's the forward pass. The differentiable physics engine that we're using in this case, called Diff Tai Chi. It's okay for the undergrads if you didn't do the differentiable assignments, you haven't seen Diff Tai Chi before. It's a special kind of physics engine that starts. The Diff Tai Chi takes over when you finish your forward pass. You write code to make this physics engine do all of this stuff, and then Diff Tai Chi says, well, the robot moved in this way, and the human wanted the robot to move like this, so it computes loss, which for our purposes today is going to be the Euclidean distance between these two vectors. So it's this area in here. This is loss. And now Diff Tai Chi is going to do a backward pass for you. It's going to basically run the simulator backwards in time. It's going to go from L to omega to theta to x, and along the way, it's going to calculate for you how all of these variables should have been different, and if they were different in that way, loss would have been less. The robot would have done more of what you wanted. Make sense? Okay. All right. So I think we got to the calculus last time. The physics engine knows that the wheel just spun at this angular velocity, and it caused the robot to move in this way. So it can work out what the physics engine knows how L will change as angular velocity changes. So let's actually plot this for a moment. We'll put omega, angular velocity, on the horizontal axis here, and L here. We'll assume that at the end of the forward pass, angular velocity was this, whatever this is. It's kind of arbitrary. It doesn't matter. Let's imagine it's 3.7 degrees per second. We're talking about velocity. So how much does the angle change per unit time? It's kind of arbitrary. Okay. That's what just happened, let's say, and Diff Tai Chi, the physics engine, knows this loss occurred. I want you to be the differentiable physics engine for a moment. Imagine that we moved a little bit to the right. Imagine that omega got a little bit bigger. What would have happened to L in that case? Assume omega is 3.7 and the red arrow occurred. The robot turned in that way. Given this theta, this x, and everything else going on in the physics engine.
Produce a bigger loss.
Would produce a bigger loss. Let's try it. Okay, we're going to make omega a little bit larger in our head, which means that the wheel is spinning a little bit, the right wheel is spinning a little bit faster. What happens to the red arrow? The red arrow is how the robot moves if that value is 3.7. If that value were 3.8, if that wheel had spun a little bit faster, how would the movement of the Bradenberg vehicle been different?
Would loss be lowered because it's spinning, so it's turning a little bit more to the right?
It's spinning a little bit. We're going to say what you just said in reverse. We're going to do a bit of the backward pass. The wheel is spinning, we're going to say it forward. If the wheel had been spinning faster, the robot would have turned a little bit more to the left than it did with this value, which means it would have traced a curve something like this, which means the difference between this arrow and what we actually wanted would be less. That's L. So if we go to the right, the physics engine knows that loss would have decreased. Loss, in this case, when I say loss, this is short form for behavioral loss. How much did this behavior lose? It lost this amount of stuff. So when we use gradient descent, which tries to minimize loss, and we apply gradient descent to robotics, we're talking about behavioral loss, which is how inefficient is the robot behaving? How far away is it from what you want it to do? Someone in the course is doing parkour. They've got a robot on one block and it has to jump onto the other block. So you can imagine the black arrow in that project, it leaves the first block and lands on the second block. That's what you'd like the robot to do somehow, jump from one block to another. If the robot jumped up and landed back on the first block, that's the red arrow. The difference between those two arrows is behavioral loss in that application. Everyone comfortable with behavioral loss? Okay, so we now know, and the physics engine knows for you, you don't have to calculate this, it knows that if omega was a little bit larger, loss would have been less. Behavioral loss would have been less. The inefficiency of the robot's behavior would have been less. The robot would more closely approximate what you want it to do. So far, so good? What about if we go in the opposite direction? What happens if omega had been a little bit less than 3.7 degrees per second? What would happen if this just happened and now we went back and re-ran angular velocity but at 3.6, a little bit slower? Everything else stays the same.
Loss would get worse.
Loss would get worse, loss would get larger, right? If this wheel spun slower, it's going to turn even faster to the right, and the difference between that new arrow and the black arrow would be greater. Behavioral loss would be bigger. Okay. let's keep going for a moment. Let's go back to assuming angular velocity is really big. So we keep turning up angular velocity. What happens eventually? If we make angular velocity a little bit higher, loss goes down. What happens if we keep making angular velocity larger?
It goes up, you overshoot.
You're going to overshoot. So loss is going to actually come down, and then it's going to start to go back up again. Okay, this picture where you have some sort of complex curve and you've got a little straight line here as an arrow should look familiar to you from calculus classes, right? So what this is, as hopefully the notation tells you, we're calculating the gradient at this point. What is the slope right here? The slope right here says at this moment in time for this value of omega, everything else being the same, the physics engine tells you you could make omega a little bit bigger and you get a better robot. So far, so good? Okay, so we have that information now. Diff Tai Chi gives it back to you. Well, for our purposes, let's just assume it's a negative number. It doesn't really matter what it is. It's just a negative slope. Okay, it gives us actually that number. It gives us this quantity, but what do we do with it? We can't really apply it to change omega because omega is something that's already calculated by the physics engine. That's what we want the physics engine to do. So we have a gradient here, but it's not really useful. We can't, this is neither a brain parameter nor a body parameter. It's some aspect of the movement of the robot. So we can't take this gradient and apply it. So let's keep going. Let's go backwards a little bit. Let's start moving back. We've got omega here. Let's go back one step to theta. Here's theta here. Okay, we know that from this gradient, the physics engine has told us that omega should be a little bit bigger. What would cause omega to be a little bit bigger? We can work upstream. Omega was set by the value of the motor neuron, and the value of the motor neuron was the value of the light sensor multiplied by theta. Possibly passed through an activation function, but we're going to ignore that. So we know that changes to theta in turn can cause changes to angular velocity. If this synaptic weight were larger, then this times this is going to cause, everything else being equal, is going to cause the angular velocity to be larger. So far, so good? Okay, I see some nods. Okay, turns out that you know that, and again, Diff Tai Chi knows that. Diff Tai Chi knows this gradient now. So let's change the labeling of the axes here. Okay, we're now thinking about how theta, what happens as we change, theta. How does omega change? So we're thinking about varying this parameter now, theta, and we want to know how omega changes. So let's imagine at this moment in time, theta happened to be 1. Remember that we set theta at random before. Let's just say we randomly set it to 1. What happens? What happens if we consider making theta, making this synaptic weight a little bit bigger? What happens to the angular velocity? What happens to omega? All else being equal.
Just increase omega.
Omega will increase. So as we increase theta, omega will increase. Look, we have another gradient, our second gradient. We're doing a backward pass. We're doing this together in class. Diff Tai Chi does all of this for you. It works backwards from the simulation that it just ran, and it's discovering all of these gradients. And Diff Tai Chi stores this in shadow variables. So for every variable that you have, there is a shadow variable. There's another variable that stores not the value of omega, but how omega changes relative to other things. Diff Tai Chi is going to give you back all of these gradients. Okay, so we have two of them, but how do we make use of them? We know that we just figured out we can't really use this one. We could maybe use this one. Turns out that if we compute the chain rule here, again, sorry, we're going back to some calculus here. The chain rule tells us that this times this times this times this gives us this gradient here. So again, if we have the two gradients, we know that we have the two gradients on the right of the equal sign there. So we can use it to compute dl over d theta now. We know how changing theta will change L. Okay, last class, tough class. You're going to have to do the chain rule in your head. A negative number, remember this gradient was a negative slope. This number is positive, it's a positive slope. A positive number times a negative number is a negative number. Look at this. We now know how to make a change to the brain of the robot. We can increase theta, and the robot will get smarter. It'll do a better job of what we wanted it to do. Alternatively, we also know that if we made theta smaller, the robot would do a worse job of what we wanted it to do. In all the discussion this morning, no random numbers, no trial and error here. We know with certainty that if at this moment, if we now run a second simulation and we make theta a little bit bigger, the robot is going to act a little bit better as a light lover. It's going to approach the light better. Question?
This is still like only on the assumption that we're changing theta by like a little bit, right? Like it's still like the
Yeah.
loss to theta is still going to be like a complex curve.
Absolutely, absolutely. You can be absolutely sure that it's a complex curve. We only know how to make a small change to theta, which brings up another question, which is how small, right? The gradient, the calculus here, the differential here just says if you change it actually by no amount, right? This is the thing that's always hard to wrap your mind around in calculus. It's saying, I know how it changes if you make an infinitesimal amount of movement in one direction. The whole deep learning revolution, autocorrect on your phone, ChatGPT, everything is based on all of this. The Achilles heel, the tricky thing is people don't really know how big this should be. How big a step can you take? You can be on the safe side and take a very, 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 very small step in this direction. Make theta, instead of making it 1, make it 1. Yes, this robot is very likely to get a little bit better at light loving. What's the problem? What's the problem in this scenario? It'll take forever. We're back to evolutionary algorithms. We're going to have to do a forward pass, a backward pass. We make a second forward pass. The robot gets a little bit better by a tiny amount. We have to do another backward pass, forward, backward, forward, backward, forward, backward. Little trade secret, Google, Meta, all those big OpenAI, all those companies, that's what they do. And they have huge data centers that are burning obscene amounts of carbon, and they're doing this, making very small steps and just doing lots and lots and lots of updates. It's a conservative and safe way to do things, but it's an extremely expensive way to do things in terms of compute, carbon, time, you name it. Yeah? So gradient descent is not perfect. It's got its drawbacks as well compared to evolutionary algorithms, but there you go. Okay. This lecture is not just about gradient descent, it's about optimizing bodies and brains. I was about to say evolving bodies and brains, no more evolution here. So the cool thing in Diff Tai Chi, among other things, is you can keep going. We can keep going backwards, and we can keep applying the chain rule. Again, we can, and Diff Tai Chi does this for you, so that it turns out that not just the neural parameters like the synaptic weights have gradients associated with them, but Diff Tai Chi also calculates morphological parameters for you. So in this hypothetical situation, if you coded up a Bradenberg vehicle in Diff Tai Chi, it would, among other things, give you back a number here, a floating point value, which might be positive, might be negative, telling you if you change x in the way that it says to, you're guaranteed to make a better Bratenberg vehicle. So you can take some of these gradients. We can take this one, and we can take this one. We can apply it, change the body of the robot by a little bit, change a part of the brain of the robot by a little bit, do a second forward pass, and the robot's going to get better at photophilia. Any questions about any of that? Yes?
I guess my big question, when we started even with Omega, like we're reasoning through the process saying, well, we know that if you spin the wheel a little bit faster, it'll turn more to the left, but that's not baked into the simulation. So how is Differential actually coming up with
Good question.
those initial gradients?
Okay, good question. So I'm hiding that detail from you. We've gone down pretty low. Let's go one level deeper. So let's open up the hood. And how is Differential doing all this? In Differential, I've said it's a differentiable simulator. It's not actually that. It's a differentiable programming language. In Differential, in this language, it looks a lot like Python. For those of you that did the differentiable assignments, you will have seen that already. It's a differentiable programming language. In that programming language, you code up whatever you want. In our case, we code up a physics engine with sensors and synaptic weights and wheels or rotational joints and light sources. And when you code it all up, among other things, you put in equations that dictate how the position of the robot changes over time. Remember, A is acceleration, and from acceleration, which is change in velocity, velocity is change in position, you can work backwards from A to change in position. This is a function of the forces acting on the robot and the masses of the parts of the robot and so on. What does Differential do? It takes the derivative of every equation you type in your code, takes the derivative of it, and can use that derivative to calculate the gradients. That's the trick. As long as all the equations you put in are differentiable. This one is differentiable. So as long as you create a physics engine in Differential where all the equations describing the physics in the physics engine are differentiable, Differential will take them, differentiate them automatically. It's known in the business of machine learning as Autodiff. It automatically differentiates all your functions for you and uses those derivatives to derive all of the gradients. Okay, I'm still hiding how exactly it does all of that. If you're really interested, go read up on the docs on Differential or have Claude explain it to you. That's as low as we're going to go today. All good? Okay, all right. For sitting through a little bit of calculus, a little bit of a reward, you can get out your devices and you're going to run this code that we just talked about on your phone. Actually, not quite on your phone. We're going to run it on one of Google's data centers. I apologize, that's very small to see. Can you read that in the back of the room, or you need me to zoom in? Can you see it? Okay. All right. So if you type in that bit.ly link, it'll take you to a Google Colab. Sorry, let me fix that. Hold on. Whoops. There it was. Better? Everybody arriving at a Google Colab? Okay, if you haven't worked with Colab before, what is it? It's basically a web page. You can type code into that web page, or alternatively, you can run code that's already sitting in that web page. I've already written some code for you. It's already sitting in that web page. Okay. All good? Okay, if you've gotten that far in Colab, you should be able to see the 3 horizontal bars at the top left. If you click those 3 bars, there'll be a drop-down menu. In the drop-down menu, tap Runtime, and there will be a submenu that comes up. Oops, sorry. This is tricky to navigate. There we go. Tap Run All. When you hit Run All, it'll take the code that's in the Colab that I wrote for you. It'll send it to one of Google's data centers, and it will run that code on your behalf on the data center. Hands up if you've gotten this far. Okay, great. Okay, let me jump back then to here. What's going to happen? Wait a minute. Literally wait 1 minute. And at the end of that minute, you should see something that looks like, oops. Oh. Why is this not running? Okay, I'm not sure why this video is not running, but it should be a relatively familiar video at this point. You will get some robot that's made up of a bunch of gray circles. These are the body parts of the robot connected together by springs. The springs are represented by the black lines. Wherever in your video you see a blue line, that's the spring being shortened by a motor. And wherever the spring is red, that's the spring getting longer and being pushed or elongated by a motor. In the top half of the video, which hopefully the code will download onto your device, you should see your robot moving relatively randomly. And in the bottom half, you should see a slightly deformed version of your robot, hopefully moving further to the right than the random robot in the top. Anybody seeing a before and after? Yes. Okay, I see a lot of smiles, common reaction. Usually it's not very efficient. What's going on? In 1 minute, that code has done 10 forward passes and 10 backward passes. Forward, backward, forward, backward, forward, backward. It saved the state of the robot at before the first forward pass. What was the random positions of all of the circles and the synaptic weights, which are driving the elongation and the contraction of the springs. And the contorted version of your robot you see at the end is a visual demonstration that Diff Tai Chi has moved these positions a little bit. It's tuning the shape of the body over these 10 passes, and it's also tuning the synaptic weights of the neural controller, which you can't see. So you now have a partly optimized robot in terms of its body or brain. Okay, you can go play around with that demo if you like. If you run it again, you'll get a different robot and a different result. It's basically not that different from what we just talked about for the Bradenburg vehicle. Any questions about any of that? All good? Okay. All right, we are going to stop lecture 23 here and switch to the final lecture on Xenobots. We got 40 minutes for this lecture. I'm going to talk a little bit about Xenobots, and because you're now all experts in evolutionary robotics, we're going to go under the hood to see the technical aspects of Xenobots, how this all works. If you're not yet tired of my voice, next week I'll be giving one of 3 of this year's University Scholars Lecture. It'll be Tuesday afternoon. You're all going to be exhausted because we will all have been here very early Tuesday morning. I will be tired too, but anyways, I will be giving a talk on computationally designed organisms. This is for the entire university community, so it's going to be at a very high level. I'm not going to talk about Diff Tai Chi and evolutionary algorithms. I will talk about some of the wider implications of this technology if you're interested. It's Tuesday afternoon, 4 to 5 in the Waterman Building in the Memorial Lounge, and there'll be yet more free food for you starting at 5 o'clock. So feel free to stop on by. Okay, Xenobots, here we go. Okay, let's rewind the tape a little bit to 2006. You'll remember that we talked about this robot. This was some of my postdoc work on injuring robots and trying to figure out ways for them to recover from their injuries. A few years after that, in 2013, a couple of biologists at Tufts University, Doug Blackiston and Mike Levin, did kind of the same thing, but with tadpoles. Okay, so what did they do? They intentionally injured a tadpole. I would love to say that no frogs were harmed in the making of this video. Not true. In this case, they took an early frog embryo, and from that frog embryo, under the microscope, Doug Blackiston, who's a very accomplished microsurgeon, took from the embryo, mechanically extracted with very small tweezers, some eye precursor cells. These are a type of stem cell. Maybe you'll be able to see a little bit better with the lights off. Eye precursor cells, they're a type of stem cell. They can't differentiate into any cell type. They've come a little bit along the path of specialization, so they can become different parts of the eye if required, usually. And what a surprise, those eye precursor cells tend to show up in early frog embryo in about the position you would expect them to be in the embryo, which is where the eye is going to develop. They took the eye precursor cells out of the embryo at that point and attached it at the opposite end of the embryo, which a few days later, as this was starting to grow into a tadpole, happened to end up on the tail of the embryo. So they've basically rearranged some biological material in the embryo. No genetic engineering here. This is completely genetically unmodified wild-type frog. Sorry, I misspoke. There was a little bit of genetic engineering here. They altered one of the genes that caused the robot to not grow eyes at the normal place. They genetically blinded this tadpole. So this tadpole, this embryo grew into this tadpole at this moment in time. It's developing with no eyes on the front. But the eye precursor cells that had been moved a few days earlier were starting to develop into something back here, this black circle. As this tadpole continued to grow, clearly this surgical rearrangement that happened to the embryo didn't kill it. It grew, and what happened was an eye started to develop on the tail. Not only did an eye start to develop here, but the eye started to send out synapses, started to send out nerve fibers that attached to the notochord or the nervous system of the frog. And as the tadpole grew into a frog, as you probably know, it retracts its tail. So the tail retracted and the eye ended up in the butt of the adult frog. God bless science, the things that you can do. So this turned out to be a perfectly healthy adult frog, except no eyes on the front, one on its butt. And when you waved some food from behind it, the frog would turn around and eat the food. It was a functional eye. Wild, right? So this lab, the Levin lab at Tufts, has become since that time increasingly famous for demonstrating something that we all learned in high school biology is wrong. Frog DNA leads to adult frog. Human DNA leads to humans. It's obvious, right? Turns out that's not true. Mostly wild type frog DNA, as I mentioned, there's been a little bit of changes to genetically blind this tadpole. Turns out that frog DNA can also encode for other things, like a frog with a functioning eye on its butt that it can use to find food. Interesting. Okay, what do you do with that fact? I don't know. But around this time, Mike Levin at Tufts reached out to me because he had read our science paper about our injuries that we visited on robots and the robots recovering. So together, Mike Levin and I put in a proposal to try and bring these two technologies together. Bring them together how? Well, if you can if a human can reach in and rearrange living tissues to produce something that's kind of like a frog but kind of not, maybe there are other rearrangements that can lead to other things that are not frog, are kind of a frog and kind of not. What other rearrangements are possible? So we propose to receive some funding to replace the human-dictated rearrangements with AI-dictated rearrangements. Can the AI come up with a way to rearrange living materials to produce a useful autonomous thing? I don't know what you want to call this. Produce a useful autonomous machine organism that does something useful on a human's behalf. Sound familiar? Okay. So we started on this project back in about 2017, and at that time we were working on soft robots. So on a Zoom call, when we first got the funding, we showed our biology colleagues at Tufts this video. We were explaining that we had AI, which is our evolutionary algorithm, that could take cubes and figure out how to rearrange those cubes to produce adaptive robots. And by the way, you can injure these robots and they can regrow. Basically what we talked about in lecture 22, evolving the bodies and brains of soft robots. Doug Blackiston, the microsurgeon who rearranged the frog tissue, was on that Zoom call. The next week, on the next Zoom call, when we were trying to figure out how to work together, he showed us this image. I remember the Zoom call, there was complete silence on the call. Us computer scientists were looking at this, and obviously it looks like we had showed the biologists the week before, but I don't know what this is. Doug explained that what we were looking at is something that's immersed in room temperature fresh water. It's just floating in a petri dish under the microscope, and it's about 2,000, 3,000 frog skin cells all smushed together. So Doug, very accomplished microsurgeon, has got very, very steady hands. He took now not precursor cells from frog embryo, but skin cells. That tadpole, that embryo was perfectly fine. It just regrew those missing 2,000 skin cells. And he smooshed them together under the microscope in the shape of the four-legged robot we'd shown the Tufts folks the week before. Okay. Again, we didn't really know what to do with this information, but now our wheels were turning. Right? So this was basically a sculpture made from frog cells of a robot. It wasn't a robot itself. So you can guess, once we understood what we were looking at, what us roboticists asked the biologists next. How can you make it move? That's literally what we said. Exactly. So Doug started to think about this and eventually figured out that he could extract from early frog embryo frog skin cells, which, like your skin, is passive and soft. It can be pulled and pushed by other bits of biological material attached to it. He also figured out that he could extract from early frog embryo frog heart muscle cells, known as myocardiac tissue. And frog heart cells, like your heart cells, these are little cells that will spontaneously increase and decrease in volume. We talked about central pattern generators in this course that have this sort of metronome. It turns out that heart cells have something kind of similar. They tend to sort of increase and decrease. And if you put frog heart or human heart cells together in the shape they expect, which for the frog is a frog heart, for us it's a human heart, somehow those heart cells talk to each other and all start to increase and decrease in size in synchrony, together in phase. That's very good news for all of us because that results in a hollow chamber that decreases in volume and increases in volume, and that chamber sucks deoxygenated blood in and pushes deoxygenated blood back out again, acts as a pump. So we got a whole bunch of things, little tiny pistons, little heart muscle cells that could increase and decrease in volume. And so Doug, the microsurgeon, said, those are the two things I can liberate the easiest from frog embryo. Are they useful to you? Can you use the heart muscle cells as motors? And remember, we had just finished up helping out with this project on soft robots. We said, absolutely. We know that if you put a bunch of things together that increase and decrease in volume, in the right distribution and in the right 3D geometry, that thing will move from left to right. So far, so good. I'll pause for a moment. Questions, comments? Anybody want to run out of the room screaming? All good? Okay, let's keep going. Okay, so what did we do? So Sam, my then PhD student, also a former student of this class, went back to Voxcraft, the soft robot simulator that we talked about last time. And instead of 4 different kinds of voxels, which is what's in Voxcraft by default, he took those 4 out and put in just 2. He put in the light blue voxels here, which are going to represent the passive soft skin of frog. Those light blue voxels are attached to their neighboring voxels with rubber beams, and the stiffness of those beams is about what you would find in frog skin. So he's kind of tuning Voxcraft to be a little bit more frog-like. We're creating sort of a model of 2 different types of frog cell. Okay. Then he created a second type of voxel, which is the red-green voxel. Red-green voxels are going to increase and decrease in volume, like the red and the green voxels that we talked about last time. But the red-green voxels, they get bigger and smaller, but they ignore the phases of the other red-green voxels. So I'll show you visually what I mean by that. Okay, so first thing, hopefully you can notice, you can see the light blue cubes being pulled and pushed by the red-green voxels. You'll notice the red-green voxels getting a little bit bigger and a little bit smaller, but you'll notice that all the red-green voxels, they're not all getting red at the same time, and they're not all getting green at the same time. They all have their own randomly set phase offset. Why? When we were starting to build these biophysical details into Voxcraft, we asked Doug, we said, what happens if you take frog heart cells and instead of putting them together into a frog heart, you rearrange them into this shape, this shape, this shape? What happens if you put them in arbitrary shapes? Will those frog heart cells in this new situation, will they still figure out how to synchronize, or will they not synchronize? And Doug said, I don't know, nobody's done this before. So we took a conservative stance, meaning we decided, let's assume they don't. Let's assume that when you rearrange them, they no longer know how to convince their neighbors how to do what they're doing. They're just going to increase and decrease in volume at their own phase offset. Yeah? Okay. Once we had that built into Voxcraft, then we went back and wrapped an evolutionary algorithm around this froggy Voxcraft, Frogcraft, if you like. And that evolutionary algorithm does what we've done before. It's hyper neat. It evolves populations of CPPNs. Each CPPN drops voxels inside the empty cage at different positions. And it paints one of 2 colors onto each voxel, blue or red-green. Here are 3 random phenotypes resulting from 3 random CPPNs in the population. In this experiment, Sam had a population size of 100, so I'm showing you just the first 3 of the 100 random CPPNs. So far, so good? So we're now doing something hopefully that's quite familiar to you all. You've done this all now a couple times. The fitness function, as always, is move from left to right. As you can see, none of these 3 are doing a very good job at moving to the right. You can't see it, but it turns out that this one down here moves 1 millimeter further to the right than these other two do. So the CPPNs that produce these two robots are deleted from the population, and the CPPN of this surviving robot, we make a randomly modified copy of it and repeat and repeat and repeat. Voxcraft is a pretty computationally expensive simulator, and we're using a computationally intensive optimizer, an evolutionary algorithm, not gradient descent. So it took us about, I'm going to skip over this for a moment. It took us about 2 weeks running on UVM supercomputer, but at the end of those 2 weeks, we got back this solution. Okay. As robotics experts at this point, you should be able to make a few interesting observations about this particular solution. What's happening here?
The red-green cubes are kind of alternating mostly in phase now.
It might look like they're oscillating in phase. We always set the phase offset at random. So these ones are just as out of sync with each other as the random ones that you saw.
So if you were to if you already had this body and then re-randomize the phase offset, would it have the same behavior?
Not the same, but it still moves more or less to the right. So this is something we haven't seen before. You've got a robot, a frog, still don't know what it is. You got something that has a bunch of actuation inside. That actuation is random. You got this desynchronized actuation, and yet there's more or less non-random behavior. You have something that moves more or less in a straight line to the right. How is this possible? Took us a few weeks to figure this out as well after we saw it. There's actually a few things going on here, and they're things that we've talked about in this course. Ideas?
I mean, if the behavior is like cyclical, then even if there is a phase offset, couldn't you just like expect it to behave relatively similarly?
Okay, good question. So you mentioned the behavior is cyclical. So if we look at an individual red-green voxel, and we plotted the volume of that voxel over time, it is cyclical. So heart cells on their own, in a heart, out of a heart, they have something like a pacemaker function. They tend to get bigger and smaller in regular oscillations. So if you put together a bunch of them that have different random phase offsets, are they going to always produce non-random motion? Are they going to move in a particular direction? Maybe, but why should it be to the right? Maybe if you reset the simulation and ran it again, maybe it would move to the left, or turn to the left, or turn to the right. This particular simulated robot does not. It always moves to the right, just sometimes a little bit more or a little bit less. Why?
My question would be, how much work is gravity doing? It looks like you essentially built a four-legged robot and just chopped off one of the legs, so it's kind of just like continually falling towards the direction that...
Now we're getting somewhere. So is gravity doing something? Probably. It ironically has come up with something that kind of looks like the four-legged robot with the front kind of cut off. What's going on here? What tends to make this thing always move to the right, regardless of the phase offsets of the red-green voxels?
It's like phenotype, like it's geometry, it's just like, it's slanted.
It's geometry, it's slanted. So the answer has something to do with gravity. It's pulling it down. It's slanted forward. You put those two things together, what's happening? What are the forces that are causing it to move to the right? You got to draw on your physics engine expertise now. Look at the red-green voxels. How are they pushing on the ground? It's a little hard to see. Are the forces of the red-green voxels pushing more or less straight down on the ground? Not really, right? Generally, they're pushing down and to the left, which creates friction and a reactive force. For every force down and to the left, there's an opposite and equal force up and to the right, causing this thing to kind of shuffle to the right. So, as always, our evolutionary process has started to exploit details of the physics engine, in this particular case, in our favor, contributing to the behavior that we want. Okay, there's another aspect, there's another interesting feature of this particular robot. You'll notice that all the red-green stuff is on the bottom and all the light blue stuff is on the top. The blue stuff is actually doing something. What?
Is it weighing it down so it doesn't come off the ground?
It's adding some weight to make sure that those forces pushing down and to the left help push it to the right. So they're adding to the weight. They're passive, and you can see them kind of being pushed and pulled around passively by the red-green voxels. Where have we talked about passivity before? Passive motion, passive dynamics. In walking. This is a form of passive dynamic locomotion, but very different from the one that we have, where our legs passively swing. These light blue voxels here, they're kind of absorbing all of the forces that are actually pushing up and to the right on them. The red-green voxels are pushing down and to the left into the ground, causing a force up and to the right, which gets absorbed into this blob of skin on the top. And this blob of skin is kind of averaging all the little up and to the right impulse forces that are impinging on that blob and performing kind of an averaging out of all of these little differences. So if you restart this robot again with different phase offsets, this blue stuff absorbs and averages them out again so that little details of these phase offsets don't affect the behavior of the robot too much. This is my favorite video so far from my career because it kind of summarizes a lot of the things we've talked about in this course and a lot of the things that I love about evolutionary robotics. Okay, all right, so we've got this simulated robot. So we got very excited at this point. This was in about 2018, a year into the project. We had something that consistently walked along the bottom of the simulated Petri dish. And it's made from two frog cell types that our microsurgeon at Tufts said he can extract from actual frog embryo. So you can guess now what phase 2 of this project is. What are we going to try and do next?
Construct it with the frog cells.
We're going to construct it with actual frog cells and hope that we cross the gap, right? Here we go. Sim to real. Okay. So let's start. We've got sim. We've got this robot here. We sent Doug, the microsurgeon, literally this video. That's it. That's what he got. He puts on his classical music on his headphones, relaxes, steadies his hands, and got to work under the microscope. I'm going to turn off the lights again so you can see a bit of what's going on here. Each of the big circles that you see here, these are basically frog eggs. These are really, really, really early frog embryos. And he's injecting a little bit of a fluorescent dye reporter. It's something that lights up fluorescently and says there's skin stem cells here and there's muscle stem cells here. This is like a sculptor who goes to the quarries outside of Rome, and the quarry master says, there's some marble over here of type X, and there's a little bit of marble over here, and the sculptor says, I want a bit of that, and I want a bit of that. Same idea. So Doug is trying to figure out where to go into the eggs to pull out the stem cells that he needs, which are going to mature into the 2 Lego bricks that he's going to try and use to build this robot. Okay. Some other gooey stuff that he's got to do. He's going to remove the vitelline membrane. So there's a little bit of sticky egg sac stuff around the eggs. You'll notice here that he's looking under the microscope and he's using microforceps. So this egg is about actually about 2 millimeters across. So 3 or 4 poppy seeds stuck together. You can see why you need very, very steady hands to do this work. Okay. Once he's done that, he's going to remove what are called the animal caps. It's the general part of the egg in which those stem cells exist. He's starting to get to the good stuff, the stuff that he wants. You're watching this. This is not a sped up or slowed down video. This is in real time. So very, very steady work here. Here in this video, you see 1, 2, 3, 4, 5, 6 animal caps, things he's just cut from 6 eggs, and he's going to remove the ectoderm, some of the outer part of the skin that he doesn't want, and he's getting at dissociated inner cells. These individual white dots that you see now, these are individual frog cells, very, very tiny. You wouldn't be able to see them with the unaided eye. And some of these cells are the ones he wants, the ones that are going to turn into skin and that are going to turn into heart. He's now extracted the ones that he wants, and he's sucked them up into a syringe, a tiny syringe. He's sort of vacuumed up all the dissociated cells, and now he's plunging the syringe and pushing or ejecting the individual frog cells, and they're settling into this big black circle that you see here. This is a little concave well in the bottom of the petri dish. So this is going to be his surgical arena. This is where he's going to do some of the actual work. I'm going to skip this one. This video is still a bit of a mystery. These are the dissociated cells sitting in the well, and you can see they kind of move a little bit on their own. Cells are covered with lots of little doohickeys on their surface, some of which the cells can actuate. They can move around. So somehow these cells are kind of moving around on their own, and you can kind of see that they don't like to be on their own. Now, whether this is just some weird artifact or whether that's actually scientifically true, that the cells themselves are like individual robots, and what we're looking at is something like swarm robotics. They're exhibiting collective behavior. They're trying to come back together. Not clear, but it looks like maybe they are. So this would be like if the quarry master gave the sculptor a whole bunch of marble type X and a whole bunch of marble type Y, and all these little bits of marble didn't like to sit by themselves. They kind of started to come back together again. My colleague at Tufts, the biologist, he refers to this as building with agential materials. If we're going to build robots from living materials, they have their own agency. They seem to be acting as if there's things they quote unquote want to do. It's a very different exercise in robotics when you're trying to build robots out of parts that kind of have a mind of their own. Okay, so we get this at the end. Doug has this purified set of about 2,000, 3,000 cells, and these cells really don't like to be alone. They're adhesive, they stick to their neighbors, and they kind of pull inward a little bit, all of them, which results in one big sphere of stem cells. Once Doug has this purified ball of frog cells, he reaches in with his microsurgical devices again, and I'll skip ahead a little bit here. Here he is on the right-hand side. There's a micro-cauterization device. This is like a little teeny tiny car cigarette lighter. It's a very, very hot wire that cauterizes or burns away bits of this ball that he doesn't want. And you'll notice that he's burned a little cross into the bottom of this ball, which is the beginnings of the 4 little legs that you see here. Okay, let me skip ahead a little bit further. Now you can see he's sort of grooving in this cross and removing or making these depressions larger and larger until he's getting something that's starting to look more or less like the thing on the left. At the very end of this surgical procedure, he's going to take this thing and turn it right side up and put it on the bottom of the Petri dish, and he's going to have this thing. So what I've shown you here is the surgical procedure that he conducted to build actually this robot, not the one that we evolved. I wanted to show you the simpler surgical procedure for a moment. I'll pause here for a moment. Take any questions if there are any. I'm going to turn the lights back on. I'll explain how he threads in the heart cells in a moment, unless there's any questions. So far, so good? Okay. All right. So this is just with frog skin, easier. So when we sent him the one with the heart muscle cells on the bottom and the skin cells on the top, he did something slightly different. The first part of the surgical procedure was the same. He extracted the stem cells that he needed from the embryos, and then he filled up 2 syringes, one with heart cells and the other with skin cells. And then he watched our video and he saw that there was supposed to be a little bit of mostly heart in the bottom. So he squirted heart stem cells into the bottom of the well, and they kind of settle into this ball of muscle. He kind of pushed it down and flattened it into a patty, and then ejected a bunch of skin cells on top, which also tried to form up into a ball, squished that down into a patty. So he's kind of making a sandwich. He's layering heart, skin, skin, heart, skin, heart, skin, heart, skin in the distribution that we want. And then at the end, he's got more or less the right distribution of heart on the bottom, skin on the top, and then goes back to this final part of the surgical procedure of scraping away what's not needed to get more or less the 3 geometry of this robot with 2 back stubby legs and is sort of lying on its chin on the bottom of the Petri dish. The result of that surgical procedure was this thing that you see down here. We're looking at this Xenobot in an aerial view. We're looking from directly above. So there's one of the back stubby legs sticking out here. You can't see the other one. I'm going to play this video now, and we're going to see them side by side. You'll notice the Xenobot in the bottom that's bouncing a little bit. That's the result of the heart muscle cells spontaneously increasing and decreasing in volume. You'll notice that obviously we didn't get the 3D geometry exactly right, and the distribution of heart and skin is also not right. It's not perfect. It's not perfect, but pretty close. Yeah. The question is, how close is it? Is the 3D geometry and the distribution of cell types dictated by the evolutionary algorithm actually doing something here? It's the exact same question you're trying to tackle in your final project. So to prove that the left to right motion we got here was not the fact that the Xenobot happened to sit on a table that was canted to the right and everything just kind of slides down to the right. We wanted to isolate the rightward movement back to this particular cause, 3D geometry and cell distribution. The way we did this was to take this Xenobot, turn it upside down, and put it back into the Petri dish. Not that complicated. If 3D geometry and cell distribution matters, if you disrupt 3D geometry and cell distribution by putting it right side up, it won't move. An alien that comes to our planet and sees a bunch of us moving like this, grabs one of us and puts us on our heads and sees that we no longer move like this, the alien can conclude the reason that these things move in this way is because of the way they're put together. It's a result of their physiology. Make sense? Okay. So let me skip ahead a little bit here. Okay, it's not the same Xenobot. Doug made another one, but it does more or less the same thing, goes to the left to the right, turned it upside down, exact same Xenobot, stops moving. We did this a bunch of times to prove that this is actually a result of an evolutionary algorithm. These videos were shot just before Thanksgiving 2019. My PhD student Sam came to my office on the other side of this wall, showed me these videos, and this happens very rarely in one's career, but this was one of those moments where we're like, oh my gosh, this is actually going to work. You can make robots out of biological materials rather than inorganic materials. It's a very exciting personal moment for me. Okay, that was 2019. We've only got we got 5 minutes left, so let me skip ahead in the story. We wrote up all this work. It got published in January 2020. Not surprisingly, CNN and New York Times, the BBC and the New Yorker got very excited and terrified by this idea of making frog bots. We got a lot of media attention, which was fun, but more importantly, it led to subsequent funding to allow us to continue this work to this day. We've continued this work, and there's now a lot of different lines of research. One of them is swarm robotics. A millimeter-sized frog bot is never going to do a huge amount of useful work for anyone, but a very large number of millimeter-sized frog bots might. So we put a bunch of them together that had just been evolved to move, nothing else. And if you sprinkle microplastics into the petri dish, they tend to push the microplastics into piles. I don't know that we're going to solve the microplastics problem that we face on this planet anytime soon, but a proof of concept that maybe these new things could act like Roombas. Even if all they do is move around and clean up stuff that they find in their environment, maybe they could be useful. Hopefully they could clean up stuff in places that are hard to reach, like the bottom of Lake Champlain or your bloodstream. These are frog skin cells. I would not recommend injecting them into your bloodstream anytime soon. Since we published the work on the Xenobots, we've now developed anthrobots, collecting human cells from humans, putting them together, and long story short, they do more or less what Xenobots do. They can also move around. Maybe in the years and decades to come, folks can figure out how to turn them into useful medical devices. At the moment, this is still basic science. Who knows, but kind of fun to think about. Here's a little robot. Here's a Xenobot that was evolved to move. And in the bottom half of this video, it looks like the Xenobot is interested in this small pellet in its environment. We did not evolve any sensors into this robot. There are no neurons inside these Xenobots. They have no brains. But this thing is acting like a Bratenberg. It looks like it's interested in this thing, at least for a while. And when it gets near to it, it turns towards it. How is that possible? Still a mystery. We don't know. But we do know that this thing is made up of about 2,000 cells, and biological cells themselves are fantastically complex robots. The surface of every cell has thousands and thousands of sensors on it, and thousands of different little micro actuators, ways of moving. So it's possible that these 2,000 cells have spoken to their neighbors and coordinated their behavior in a little bit, in some way. Okay, we got 3 minutes left, so let me now pull back a little bit. I've shown you in this last lecture Xenobots, which have raised more questions than they've answered. Along the way in this project, there's a lot of interesting biology questions now. This particular Xenobot is made up of genetically unmodified material. So frog DNA apparently encodes for normal adult frog. It also encodes for this under the right environmental conditions, which include a microsurgeon intervening on that material, what else is possible? I don't know what the future holds for robotics, but I'm excited in the years to come as we learn to build robots from a wider and wider range of inorganic and organic materials. We saw some of these inorganic materials in the soft robotics lecture, some of the biological materials that are possible now. I'm also excited about trying to build all of these autonomous machines from more and more materials to be autonomous and useful and safe on our behalf. As we saw in the open questions section of this course, that's a bigger question, a bigger unanswered question. I hope some of you have found this course interesting and possibly will pursue robotics. So I think with that, we're going to end the lecture portion of this course. It's been an honor and privilege to share evolutionary robotics with all of you. There is one final quiz tonight, and then I will see you all back here very early Tuesday morning at 7:30 a.m. I'm very much looking forward to seeing what you came up with in your final project. Okay, thanks very much, everyone. See you on Tuesday.